All right, today we're diving deep into the science of the psychedelic experience. But we're not talking about culture or therapy. Nope, we're going right to the source. We're talking chemistry. We're going to look at how these incredible molecules are actually built and what they do to our brains on a fundamental level.
And it's really the central question, isn't it? How can this tiny little thing, just a collection of atoms, completely change our consciousness, our perception of reality? Well, the answer is all in the chemistry. It's this amazing dance between the shape of a molecule and the machinery inside our own cells. So let's start with the basic blueprint.
Okay, so the first thing you gotta know is that even though they all have different effects, the classic psychedelics are all built on a shared foundation. They all follow this one common chemical blueprint, and that's what lets them work their magic in our brains.
And there's a cool scientific name for this blueprint, a pharmacophore. The best way to think about it is like a skeleton key. It's the most important part of the molecule's shape, the part that lets it fit into a very specific lock inside the brain. It's not the whole key, not the fancy handle, but it's the part with the teeth that actually does all the work.
And here it is. This is that skeleton key. The basic psychedelic pharmacophore, you can see it highlighted here in blue, has three essential parts. You've got an aromatic ring, then this nitrogen group called a basic amine, and, this is crucial, a two-carbon chain linking them together. That exact spacing, that two-carbon link, that's kind of the magic ingredient. It creates the absolute perfect shape to interact with our brain's receptors.
So, starting from that one basic blueprint, psychedelics branch out into two major chemical families. Think of them like two different brands of keys, but they're both cut from the very same master design. This chart here basically shows the chemical family tree. You can see our basic pharmacophore right at the top, and then branching down from it are the two main lines. Over on the left, we've got the tryptamines, and on the right, the phenethylamines.
Okay, let's zoom in a bit. The real difference between these two families is in that aromatic group we just talked about. On the left, you've got tryptamines, like psilocin, that's the active stuff in magic mushrooms, and they have this double-ring structure. Then on the right, you see phenethylamines, like mescaline, from the peyote cactus, and they have a simpler, single ring. It seems like a small change in shape, but it has huge consequences for how they actually work.
So, what about the most famous psychedelic of all? Where does LSD fit into all this? Well, LSD is a real wild card. It's not really in one family or the other. Instead, it's this fascinating hybrid that grabs the most important features from both. And you can literally see that hybrid nature right here. Look at the core structure, the part in gray. That's a classic tryptamine. But then, look closer. Tucked inside that big, rigid structure is the phenethylamine part, highlighted in yellow. This one-of-a-kind combination is a big part of why LSD is just so incredibly potent. So, we've got our key, the psychedelic molecule. Now we've got to talk about the lock that it fits into. Because all these different molecules, they all head for one primary target in the brain, one specific protein that works as the main docking station for them.
And that lock is the 5-HT2A serotonin receptor. And believe me, the evidence for this is just overwhelming. In studies, if you block these receptors with another drug, the psychedelic effects of something like psilocybin or LSD just get shut down. They even engineered mice without this receptor, and they just don't respond to these compounds at all. So the main takeaway is simple. No 5-HT2A, no psychedelic trip.
And this right here is what it looks like. This is the complex protein that sits on the surface of our brain cells, just waiting. So when a psychedelic molecule comes along, our little skeleton key, it slots right into a pocket on this receptor. And that one simple action kicks off a whole cascade of events inside the cell.
So what happens after the key turns in the lock? Well, binding to the receptor is really just the beginning. It triggers this complex chain reaction, a kind of cellular domino effect that ultimately changes how the entire neuron functions. So what does this chain reaction actually look like? Well, it all starts when the psychedelic molecule, which scientists call an agonist, finds its target on the cell surface. So here's the first domino. Our psychedelic, the agonist, docks with that 5-HT2A receptor on the outside of the neuron. This simple connection causes the receptor to literally change its shape, and that physical shift sends a signal to the inside of the cell. That signal then wakes up a helper protein that's just waiting inside the cell membrane. It pops off and carries the message deeper inside, kind of like a relay runner passing the baton. That's the second domino falling. Next, that helper protein turns on an enzyme that works like a tiny pair of scissors.
It snips a molecule in the cell membrane, which then releases two new messenger molecules. So now, a single signal has just branched into two, getting ready to cause multiple effects all at once.
And here's the final step. One of those new messengers travels to a storage unit inside the neuron and opens a gate, causing this flood of calcium to be released. This calcium rush is a super powerful internal signal. It changes the neuron's electrical activity, it can even change how its genes are expressed, and it ultimately creates that profound shift in consciousness that we call the psychedelic experience.
So there you have it. From a simple chemical blueprint to a complex cellular domino effect, we've followed the entire molecular journey of a psychedelic. And you know, understanding this process in such detail, it just opens up some incredible new possibilities for the future. So let's just do a quick recap. We learned that all classic psychedelics have that core skeleton key structure, the pharmacophore. We know that this key fits into a very specific lock in the brain, the 5-HT2A receptor. And turning that key sets off a whole domino effect of signals inside the neuron. But if there's one thing to really take away, it's this. A molecule's shape isn't just some random detail. It is its function. You change the shape, you change the whole story. And that, of course, leaves us with a really exciting question. Now that we understand this chemical blueprint so well, now that we know the shape of the key and the lock it fits, what new keys could we design? Could scientists build molecules that maybe turn the lock just enough to treat anxiety without hallucinations? Or maybe create keys that can boost brain plasticity to fight depression? We're just starting to see what's possible.